Yeah. That's rough. I actually did the actual calculation, but it's close to 8,000. All right, when you guys get that picture up, I'm ready. And we'll pray before we start. Would it be difficult to put the picture I have on my screen up?
Yeah, just go airplane to, I'll stop by and do airplane.
You want me to click AirPlay? I don't even know where that is. It's under this, isn't it?
Right there.
This one.
No,
This. Okay, yeah, that's the screen sharing.
And then screen mirroring.
Okay. Apple, okay. Is it? Oh, great. All right. Let's pray together. Father, we are more than amazed, enthralled for the little bit of creation that we understand. So help us, as it's important and necessary to grasp more, partly because we can share it with others, partly because it strengthens our own determination to not doubt the Word of God, which is so beautiful and so powerful, Lord. Thank you in Jesus' name. Amen. I want to tell you about these three men. On the left, not the man, but the three names that are on there. Stephen Meyer is the director of the Discovery Institute in Seattle. He has a PhD in philosophy and a couple of other advanced degrees. A very precious guy. He's written about 4 books. I've read most of them. When I say I read, once in a while, it means that I went through and spent several hours reading something here and there, depending on how much I felt I needed to do. But anyway, wonderful Christian man. Very gentle, very informed. Such a treasure. David Berlinski is a Jew, and it's not fair. Have you ever noticed how they're so much smarter than the rest of us? I'm serious. And you know why that is? God bless them. I'm sorry? Well, yeah, God blessed them in various ways, didn't he? David Berlinski is an agnostic. Do you know what that means? Without knowledge. And what that means is, in a sense, he's like you and me. Don't you and I believe in God by faith? Is there evidence? But it's not, if you will, inconclusive. That is to say, we've never met him and shaken his hand, right? So Berlinski is this honest, very honest fellow who technically is an agnostic because he hasn't had that experience. But it's very clear to him. As you'll see him, we're going to play about a 30-second clip of him answering a question. And I think he's probably one of the 2 or 3 smartest men on the face of the planet. I've listened to him lecture numerous times. He'll get up and talk for an hour on some complex presentation and never have a note the whole time. And just quoting paragraphs and referencing. It's just astonishing. David Gallernter, this is a very interesting story. A dedicated evolutionist all of his life. He is the head of computer science at Yale. He is respected as probably the top computer science guy in the world, if you will. And he has come to the place where he says it's impossible that these things could have happened by chance. And he's become a believer. He's struggling. It's worth listening to this whole hour-long interview because all 3 of these guys make very cogent, comments. So if you get a chance, all you have to do is put in their last name, all three of them, in YouTube, and this video will pop right up. But because when I screen recorded it, the sound was bad, the gentlemen in the back are about to play it on the screen. Is that correct? Do I need to disconnect myself from the screen or anything? Okay. Peter Robbins is the interviewer, a very interesting Christian man.
Tell me if I've got it more or less right. In Darwin's time, tell me if I've got it more or less right. In Darwin's time, it was good enough to imagine that the basic unit of life, a cell, was like a little brick of Jell-O. It was an undifferentiated, quite uncomplicated thing, and you could imagine putting many, many, many of them together and getting different forms of life. Is that roughly fair?
Yeah, it was good enough for Darwin. It's probably good enough for us as well, but it's not true.
It's not
That's the big problem. The cell is an unbelievably complex bit of machinery, unfathomably complex. And we haven't understood its complexity at all. Every time we look, there seems to be an additional layer of rebarbative complexity that needs to be factored into our theories. Don't forget, the eternal goal is to explain the emergence of this complexity.
Yes.
And if we're continually behind the curve because the complexity is increasing every time we look, that eternal goal is also receding from view, not approaching, it's receding. It's becoming more and more difficult to construct a theory for that.
All right, now,
Good, thank you.
somebody give me...
Can you put me back up there? You need to go home and listen to this whole thing, and particularly listen to Berlinski several times over. He said, in fact, I've actually taken some of, go ahead, what you want to... Oh, okay, very good. Thank you. I thought I had them here, but maybe I shortened it up without them. So I'll just mention it. You heard him say, we haven't understood the complexity at all, in spite of the fact, this is one of my books on molecular cell biology. What are there? 1, 2, 3, 4, 5, 6, 7, or 8 PhDs in molecular biology that are the authors of this book. I can open this book up, and virtually anything we know about the cell, I can start to read it and maybe look at a paragraph, and it might take me an hour or two to really get my mind around everything that's in that paragraph. You know what I'm saying? It is unbelievable, the knowledge that we have. And Berlinski is not unaware of that. And what did he say? We haven't understood the complexity what? At all. Every time we look, we figured a bunch of stuff out, and then we decide to probe some more, and he said, another unfathomable layer of complexity. It is just beyond belief, friends. And he used a word I've never heard before, a barbative. It just means really big, really more. So if you get a chance to listen to Berlinski, it's well worth your time. Here's a comment he made. Darwin's theory of evolution is the last of the great 19th century mystery religions. What's he calling it? As we speak, it is now following Freudians and Marxism into the nether regions. And I'm quite sure that Freud, Marx, and Darwin are commiserating one with the other in the dark dungeon where discarded gods gather. Richard Dawkins, the most famous atheist probably today. He's very clever, very bright, very clever, can get people in a corner that are arguing the other side. Wrote a book called The God Delusion. In fact, Terry Martin's son, is it not Trevor? Tanner, stood behind this pulpit, 5 years ago, and read a paragraph from The God Delusion by Dawkins, where it's just so sacrilegious, it's unbelievable. He's calling God all these terrible names. And so people that don't want to believe in God love to hear people like Dawkins because it makes them feel good about their choice. But Berlinski, the guy that you just listened to, then wrote a book called The Devil's Delusion. You should look at it. It's pretty high stuff. He is so esoteric in his thinking that I can read a paragraph and I need to just sit down and close the book, or maybe keep the paragraph in front of me and spend a half an hour trying to make sure I understand what he said. He's just so deep. So he wrote this book, The Devil's Delusion, in answer to Dawkins. I want to take just a few minutes. There's a number of you in here that know way more than this, and my apologies, but there's some of us that need a little tutoring on the DNA. So this is a drawing, artist conception, a simple conception of a cell. And down in here in the middle is what we call the nucleus. And inside the nucleus are the chromosomes, or if you will, the DNA. If you take all the chromosomes, and I think, don't most of you know, and I'll show you a picture, the DNA is like a ladder, and it's been twisted in opposite directions, top and bottom. Are we all together on that? And if you hooked all the chromosomes together, they each have their own chunk of the DNA, it would only be about 6 feet long. But the whole thing is wrapped up in a way that you'll see in a moment, so that it's in a space tinier than almost you can put your fingers together. This isn't just a drawing. Just so you'll know, every chromosome, this side is from mom, that's her DNA, and this side is from dad. And it's not right and left, because we could have been looking at the other side. The point is that they're both there in these chromosomes, and they are almost all the same. Almost everybody in this room, our DNA is probably the same to 99.99%. Just little tiny differences here and there. But there are differences, even in moms and dads. And many of these differences are the results of mutations, which I'll talk about in a minute. And I should have used those arrows instead of my hand. Now this was published in 2006, I think it was, Newsweek edition on the DNA. It was every article in there was about this kind of stuff that I'm talking about, will be. And you can all see the twisted ladder down the middle. You can see the rungs in the ladder. And the reason that granddaughter looks a lot like grandma is because of the DNA, as you know. Here's an artist's conception that reveals something that the previous picture did not, and that is that the rungs are actually 2 half rungs. And there's actually 4 kinds of half rungs. And the people the 2 guys that discovered this structure understood very early in their work that the A rungs, there's only 4 kinds of rungs, and these are the letters that we designate with them because it's the first letter of the chemical name, but it's just think of the letter. The A rungs only hook to the T rungs. They're not shaped like this in reality, but the artist drew them that way to help you see that yellows are hooked to greens or greens to yellow. Are you all with me in the idea? And blues hooked to reds. They're not colored. They're not shaped exactly like this. There's chemical reasons for the connections. But G's and C's always hook together so you can see the red G and the blue C. Just as in the words tile and light, you can see immediately they're the same letters. Without reading the rest, what makes the difference when you and I read those 2 words? You used order. She said how they're put together. And a scientist would rather have you say the sequence of the letters, but you're all meaning the same thing. So it's the order of the 4 letters that provides the information, or the pattern, if you will, or the code to do what? Did you pick that up from this morning? It takes a gene which has the information for the machinery in the cell to make a certain protein. Are you all with me on that? And I'm actually going to show you a video of how that happens. But the idea that the gene codes for a certain protein is real critical to this. So what do? They have the instructions. I should have put that in quote, or they have the code for a particular protein. They're used by the manufacturing facility in the cell to make a protein. And this is an artist's conception. But here's one of those factories. Can you see the little black dot at the end of the arrow? Say yes? It has a name. The name's not important. Can you see that there's lots of these little black dots? Hundreds of thousands of factories in any one cell that manufacture proteins. All the proteins that that cell needs. There's the name for it. Don't worry about it. And they've got 3 arrows pointing at 3 more of them. Now here's the video. And I'm going to talk instead of the man that I'd love to tell you about, and I will tell you briefly about him. So let me make sure the sound is off. And if I can make myself press the letter K instead of the mouse or an arrow, I won't have to start the video over again. So here we go. We're inside the cell, and we're traveling toward the nucleus. The nucleus has little holes. They have a name, sort of. And there's a sentinel at each hole to let things in or out, or not let them if they shouldn't. Can you see already the DNA inside the cell? It's simplified. And in a moment, there's going to be a protein, by the way, a protein which the cell makes in order to unravel the DNA so that the 2 sides are separated. And then the same protein facilitates, I'm going to stop it. Can you see how it's cutting the rungs, cutting between the 2 rungs, everybody? You all with me on that? Please stop me if it's not clear. This gadget right here is a protein that separates, and they don't show it here. I'll show you in another video. They facilitate, wait till I get there, building the other half. So here comes, by the way, can you see these half rungs here? Yeah. So those are A's or G's or T's or C's. Are y'all with me on that? And now we're going to build, if this is a T, we're going to put an A there. If this is a G, a C, and so forth. There's actually, I'm sorry for this one tiny difference. What we're about to make is some messenger RNA. If it wasn't for the vaccine, I probably wouldn't have even mentioned this. You're all aware that the vaccine is actually messenger RNA. You all know that, right? And it's manufactured, in the case here, in the cell. And instead of hooking an A to a T, there's a different half rung that's used, which helps the body know this is messenger RNA and not just one side of DNA. It's not terribly important, but some of you know that, and you'll wonder why I didn't mention it, perhaps. So there, it's building the other side of the ladder. Are you all with me? Can you see that, everybody? And it's hooking the right ones up. It's hooking A's to T's and G's to C's, with the exception of U's to T's instead. But don't worry about that. So there's that copy, if you will. Are you all with me on that? That thing that's going off to your left? It's not an exact copy because of the U half rungs, but we consider it a copy because it has the information, and it's going to go outside now to that factory where the protein is manufactured. This is messenger RNA, or mRNA. And it got that name because it has the information for the factory to use. So it's a messenger coming there with that information. The sentinel allowed it to pass. By the way, Drs. Sarton, we've always believed that the messenger RNA can't get back inside the nucleus. A study was published less than 3 weeks ago, about 2.5 weeks ago, that in vitro, the messenger RNA is getting inside the nucleus. Pretty interesting and maybe even scary in terms of the vaccine issues. But nevertheless, here comes just because it happened in vitro on a plate. And this was liver cells that the messenger RNA was getting back into the nucleus. Doesn't mean that would happen in a living creature. So you always have to keep studying. Boy, I almost hit that arrow key, and we would have had to start all over. Whew, thank you, Lord. Now here is the factory, which is actually sitting around in two pieces until some messenger RNA comes along, and then the two pieces of the factory come together and start the process of, let me show you something. I'm going to call this a truck. And to the truck is hooked an amino acid. This amino acid and this truck are unique. This truck cannot haul any other amino acid. How many of them are there? How many different kinds of amino acids? 20. I'm mixing you up because the 4 was the half rungs. You remember from our church service, I talked about 20 different kinds of amino acids. Plants make hundreds more than that. We don't use them. We use 20 of the amino acids to make these proteins. My point is, this truck is unique to that amino acid. And the truck has half rungs on its nose, which those 3 half rungs correspond to that amino acid. You got that kind of? You'll get it some more if you don't. So the truck is allowed to park and see if its 3 are the correct 3 so that the amino acid sequence will be correct. Does that make sense? And now he's going to wait for the next truck. And can you tell that this is a different shape amino acid than that one? And this truck is unique to that one based on its half rungs. Can you see that those half rungs were different than these 3 half rungs? The order of them, right? So something very interesting happens right there. If this matches, then a chemical reaction connects the 2 amino acids and this truck can leave. Got it? Now you try to do that in the lab. You try to hook amino acids together in the lab. It is very hard. It takes some very specially chemical concentrations and temperatures. It's tough to make them hook together. But it happens in the cell just so easy. And here comes another unique truck. Can you tell the difference in the rungs? They've used a different length to try to show you they're different. Y'all with me? The length is not really the issue, but they're using that. And can you see this is a different shape than that one? So, again, the truck is checked before it's allowed for the chemical bonding and for the other truck to leave. Y'all with me on this process? So we're assembling the amino acids, unlike in the pews today when it was random, right? This is assembling in a perfect order, correct? The proper order. And so Berlinski said, unfathomable complexity. We'll let it run here for a few seconds, and then I'll show you something a little different. Now, this is the outside view. Tell me what this, I want you all to think of what it is, but don't say the answer out loud. What is this thing up here? Somebody raise their hand and give me the answer. Yes? Go ahead. Yes, it is the mRNA. How many got it right? Let me see your hands. Oh, bad teacher. You must be mixed up. How many are mixed up? Let me see your hands. I got to tell you a story. I was giving a lecture a little bit like this part in a church in College Place. And one of my friends from Academy came to hear it. And she was a straight A student back in those days at Milo Academy. And so I went to talk to her afterwards, and I don't know what I said, what do you think, or something. And she said, you lost me after the first 2 sentences. And I'm thinking, what? I was doing such a good job. Anyway, tell me where you're mixed up, because you didn't know it was messenger RNA. Let me ask you this, since you're not raising your hand. Do you get it now that that thing that's out there, that's the messenger RNA that's being read, right, to get the right sequence of amino acids? Okay. What's this thing down here? Don't say it out loud. I want you all to get the answer in your head. It's coming out of the factory. You understand that, right? See it come out of the factory? You all with me? What is that? Somebody raise their hand.
Amino acid.
Very, very close. I'm sure you're thinking correct. It's the amino acid sequence. Yes. Are you all with me? That's going to become the protein. It is not yet the protein. It's got the right amino acid sequence. And by the way, this thing happens at blinding speed. Hundreds of amino acids per second can be assembled. It's just astonishing. That amino acid sequence is taken to a folding factory. And it's funny, there are proteins in there, listen to all of this, friends, that help fold it up into a certain shape. You all with me? And the scientists have called those proteins chaperones. They're folding it up. And the protein, I said this morning, please understand this. Proteins can only do their job because of when they have the proper shape or because of their shape. So it's critical. And you just get one amino acid wrong and it can't hold the shape that it's supposed to have. Are you all with me on that? It doesn't take much to cause these genetic problems by just one. Now, here comes a truck from your left. Most everything that moves around in the cell is transported by, this is a metaphor, trucks, along little highways that are built in front of the truck as it travels. And as the truck passes over the highway is disassembled right behind it. And the reason that the highways are not in there all the time is the cell would become stiff. So you have a truck, a unique truck to carry something, and a highway is built for it, and there are genetic mistakes. There's actually flagmen, that's a metaphor, about where to turn. And there are genetic errors. There are diseases caused by a genetic error that makes, when the flagman said turn this way instead of that way. It's just, what did he say? We haven't understood the complexity what? At all. I mean, we know so much, but the amount we don't know is probably 1,000 times of what we know. And he said, you heard him say, and you'll never find anybody more appraised than David Berlinski, I can promise you that. Boy, my hand wants to press that arrow key, and then you have to start the video all over. I'm going to tell you about Dean Kenyon, a professor at UC San Francisco who wrote a book, you can look this up on your cell phone right now if you want, called Biochemical Predestination. He wrote it in concert with a Russian author whose name I don't know or don't remember. And it was the answer to the evolutionary problem, hailed the world over. Oh, we finally understand how these proteins can assemble. They have the information built into themselves. And one day, a student in one of his classes raised his hand and asked Kenyon a question. The question I have not yet been able to find, but I've heard Kenyon tell this story. And he, to his great credit, Marilyn was asking, is Marilyn here? Marilyn's son, I don't think you would mind Marilyn. Marilyn's son is a very well-respected plant scientist, and he has become an atheist or an evolutionist. Her son. I had a nice conversation for an hour with her son when I learned that his world was to study the DNA in plants. So I had a lot of questions. But Kenyon has become a believer. He went back to his laboratory and went back to evaluate what he had done, and he realized that they had made a mistake, and he is now a big promoter of creation.
What's his first name?
Dean Kenyon, Kenyon. So I'm going to not have you listen to him. It's a great story he has to tell, but I want to go to the next slide here. And, here's some audio. Am I hooked up for audio, gentlemen, back there? This is good audio. Shall we try it? Well, I'll just read what he says. Antony Flew was the world's most famous atheist. He was the son of an son of a Methodist minister. Became an atheist when he was 15. He wrote over 30 books during his life, but he was known best for his what? Arguments against the existence of God. And he was considered the world over. He was the Richard Dawkins of his time for many years. And he was from the UK. But he wrote, he was lecturing, and he said, "Has science discovered God?" And here's his last publication. Marilyn has read this book as well. There is no God. And what did he do with the no? He crossed it out. And look at his bust. What's the orientation? He was humble enough to say, "I was dead wrong," and published this book. And here is the interview. What were some of the factors that prompted you in recent years to reconsider atheism, to come to the conclusion that there's an intelligence? And here's his answer. It's been entirely these biological discoveries, the integrated complexity. Then he goes on. This is Flew speaking. If the integrated complexity of the physical world is a good reason, as Einstein clearly thought it was, of believing that there was an intelligence behind it. Behind what? The cosmos, all right? The world, the cosmos. Then this argument applies a fortiori. There's a word I didn't know before either. It just means with much more force. Applies a fortiori with the inordinately greater integrated complexity of the living world. Now, this was 20 years ago, or maybe not quite that long, maybe 15 years. And of course, what we've learned then since just makes this pale. But he says, "It seems to me this is just obvious." The argument is much stronger now. And Marilyn knows, we talked about this. In a way, he's a deist. He's just saying, well, there is a God. I don't know him, but you know what? There are other interviews of Flew. A pastor interviews him and says, "Do you think it's a personal God?" And Flew said, "Well, he would have to be." Interestingly enough. So, the deist that he is. These are Berlinski's words. We closed his talk, his little clip there, with him saying, the eternal goal was to explain the emergence of this complexity. That is to say, how did the first cell ever form? You could argue, if you had a cell with all the DNA, maybe you could tease it into becoming a fly or a frog or something, maybe. But how did that first cell form? And what did he say? The more we learn, the farther and farther away is the possibility for us to ever figure that out. Did you catch that?
The more it recedes.
Yeah, further it recedes. It's not getting closer because of what we're learning about the cell. It's becoming more and more impossible. Now this one, I want you to hear the audio. What do I have to do? The audio should work. Let's just try it and see. I have to turn mine on, don't I?
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called a nucleus.
She said most. The only cell that doesn't have a nucleus is the red blood cell, which, by the way, is a large percentage of our cells. But anyway, that's where that comes from. So there's the nucleus, just like we saw before.
The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long
I'm still plugged in. It didn't run out of power. My computer does this once in a while. Something isn't right. It's going to take 30 seconds. Any Q&A while this thing reboots? Something is scaring me here. Nothing comes to your mind?
So where do you find all this information?
Oh, I'm always looking for stuff. His question was, where do I get all this? A lot of it, these clips come from YouTube. But I've bought a number of these books that I've been talking about. But any question? Yes?
So explain a little more, what is the connection between the vaccine and the mRNA?
Oh, the vaccine is messenger RNA, which has the information, and this should make sense to you, for the cell, our own cells, to make the spike protein that's on the virus. Are you all with me on that? And the idea then is you make some spike protein that gets into your blood, and then your body, the immune system of our bodies, recognize it as foreign material and make antibodies against that spike protein. And those antibodies, when they see a virus with that spike protein, you don't have to do an awful lot to decommission a virus. So it's an elegant idea. A bunch of you must have listened to the guy that invented this and became against its use. Are you aware of that? What's his name? Robert Malone. Yeah. He's a long-time researcher on vaccinations. And it's an elegant, do you understand what I mean? It's an elegant idea. Yeah, let your body make some proteins, and you don't even have to put a virus in yourself to get some immunity. You follow what I'm saying? My problem as a scientist is you have no idea what this means 10 years down the road. None. And it might be just fine. But that's my problem. And the other thing is, you all know this, if you're healthy, your chance of dying, now we've had a couple of people in our church, I realize it's a tragic story, but if you're healthy, your chance of dying is around 1 in 1,000. Just the general population, it's 0.05% chance of dying. Half of 1%, a little less than that actually.
And the other thing is that you might introduce the spike protein from the mRNA, but that's only one little component of the immunity, whereas when you actually are exposed to the real virus, there's this whole complexity that you get exposed to.
Yeah. So, this thing has become so political and so controversial, I hate to even get into it, but it is messenger RNA. That is the vaccine. Now, they have to put that vaccine in a soap bubble. Did you know that? Well, it's actually made of phospholipids, but so that it's water soluble. And then that soap bubble, that water soluble bubble, touches a cell, and it's like two soap bubbles. They go boop, and it's suddenly inside. That's how they get the messenger RNA inside the cell. Okay. Anybody else? Comment or question? Just speak up. I think I can get back there quite quick. If I had some glasses, it would even be quicker.
I just think that it's so amazing that you have that nucleus to keep the genetic material stored away. It's like keep it in a safe. And you're careful to only let out just what's needed. You only make copies and let the copies out. You don't put the original out into the cells to float around. It's all kept safe inside that nucleus. Yeah, good point.
Okay, it even remembered where we quit. Isn't that amazing? So now I want to take you to, oh, by the way, we didn't see the end of that, did we? Yeah, let's play that again. We barely saw the start.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus
I did.
contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes.
Stop.
These genes contain the instructions for
So a gene, in this case, they've illuminated it, is from here to there. Are y'all with me? So a gene is a portion of the DNA. When the scientists by 2020 had sequenced the DNA, I didn't tell you, there are 3 billion 200 million rungs in this ladder. Did you hear me? And the scientists wanted to know the order of the A's and the T's and the G's and the C's. Are y'all with me? And that's called sequencing the human genome. That was completed in 10 years. By 2020, it was finished. And scientists are blown away still. Only 1.5% of the ladder is gene. The rest of the ladder is being studied intently. The rest of the ladder seems to be involved, and there's a lot of knowledge already, with controlling whether the gene is going to be used or not. They use a lay term, switching it on or switching it off. But anyway, let's listen to what she says about this gene. So this is another look at the very same process, but it had some features I thought would be helpful.
making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA.
Now, can you see how it has separated the ladder, like before? And there's a bunch of free half rungs sitting around, and these are used to attach in the proper order, or the proper one to the proper one, you'll see momentarily, to build what? To build the messenger RNA. Are y'all with me? You're not with me. The messenger RNA is going to be assembled right here using that side of the DNA. You could use either side as long as you know in the cell what's going on. So let's watch it happen.
Out of free bases in the nucleus.
Now do you see that A's hooked to T's and U, that's the unusual one, hooks to A and so forth. Y'all with me on this? So this U half rung is replacing a T half rung, but not the other way around, interestingly enough. There we go.
The DNA code determines the order in which the free bases are added to the messenger RNA.
Bases is a fancy term for the half rung. Are y'all with me on that? They're called nucleotide bases. Sorry for the big terms.
This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing...
This is very interesting. The other video didn't mention this at all. But a gene, a piece of the ladder, are y'all with me? Actually has what we call introns and exons. And one gene we now know, you can take that piece here and hook it to that piece there and get that one over there. One gene could code for, who knows, 100 different proteins. And we do not yet understand how the cell knows how to take which one to hook to which one. What did he say? Every time we look, there's another layer of unfathomable complexity. Just astonishing. But here she mentions it. They take an intron out and hook the other two pieces together.
And adding sections of RNA.
Get the idea? Y'all with me? Remarkable.
The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA.
Yes. Sure. You mean separating the DNA so that you can copy, use the DNA to make a copy that you take out. So you never take the DNA out of the nucleus. God just designed it so that the DNA would stay in the nucleus, but you separate it, make a copy, and then you take the copy to the factory. Because the copy it has the information to make a specific protein. I know so much has gone by, folks. You might have missed this. I appreciate her being willing to ask this question. The point is that a gene ultimately has the information for the ribosome to make a specific protein, correct? And for whatever reason the Lord had in mind, he leaves the DNA in the nucleus. So what you have to do is make a copy of it, if you will, that you take out to the factory so the factory can use it to make the protein that's needed. The proteins, remember I said this morning, there's hundreds of thousands of different proteins to do jobs in the cell. One job is making the highway in front of the thing that's got to be carried somewhere. Another job is the truck itself. And on and on and on, the jobs that have to be done. All of these proteins are little machines, and they do stuff. There's this very interesting protein. I wish I had a blackboard. I'll just draw it in the air. If you take the nerve cell in our brains and everywhere, there's a protein that stands on the edge of the nerve cell, and by changing shape, it causes 2 potassiums to be pulled out of the nerve cell and 3 sodiums to be pulled in. And that's an electrical charge now, because each of those have a charge of 1. So 3 in and 2 out, there's a net charge of 1. Are you all with me on this? And that protein, all along our nerve cells, is constantly chuk, 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 or we would never think, we would never move. It's just one example of a protein that's a machine, right? That pumps, we call it a protein pump. It pumps electricity, it's actually an ion, in and out of the nerve cell. And once the electricity is pumped out there, then you can have a wave of electricity, because they jump back inside. I show you pictures of all that, but just to give you an idea, hundreds of thousands of jobs that have to be done in the body, all performed by proteins. And they're able to do it because of their shape.
So, and I don't know if this is kind of what Nancy's referring to, but I just think about, that there's an awful lot of, that goes on just to make a protein. But maybe we're not appreciating the little things.
I'm not sure what...
All of the splitting of the DNA and making an RNA, all this stuff just to make a protein.
That's correct, yeah.
But it's like maybe we aren't appreciating what these little proteins do.
Yeah, I didn't emphasize that enough, so I just tried to give an example. There's just an incredible number of things that are done by little protein machines. Where's Lisa? Oh, there she is.
Oh, just restating to be sure I'm understanding. So the DNA is split inside the nucleus.
Yes.
Then
A portion is opened up.
Portion is opened up, half piece goes out, the mRNA, and then the little trucks bring in the amino acids that have to hook on in the right sequence.
Yeah.
And so that sequence of amino acids is then what is taken to build the protein.
Well, that is the protein once it's folded into the proper shape. Yeah, and by the way, the little truck has a name too. They call that transfer RNA. And so everything's got to be named. In fact, there's one right there. They get a little better drawing in this
In the messenger RNA to produce a chain made up of amino acids. There are 20
And you see how this truck is carrying an amino acid? You all with me on that? A little different drawing than the other one.
different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time.
See how they match? You all with me on this? To make sure it's the right truck and has the right amino acid.
Each triplet is read. A transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids.
You remember the other movie? We were looking at the growing chain of amino acids, right?
Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So in my classes, I try to get people to always answer me when I say proteins work because of their shape. It's more complicated than that. It's also electrical signature. So that the plus on the thing that's going to be worked on by the protein matches a minus on the protein. You all with me? Very complex electrical interplay as well as shape. So it's just another layer of what? Unfathomable complexity. Okay, enough of that. Now I want you to go with me to my dear friend who we lost recently, Robert Gentry. And this is where we need to do a little nuclear physics. Robert Gentry found in granite tiny, tiny, microscopically sized rings, which he realized were created by, really, it's a sphere. But when you cut the granite, what does the sphere look like? A ring. Y'all with me on this? And he realized that these rings were caused by radioactive particles emanating from the center of the ring and going a little distance before they crashed and burned. And when they did that, they make a little mark. And when a bunch of them do it, they make a ring or a sphere. Y'all following me? Ask me some questions that I didn't make it clear. It's a sphere in the granite, but when you slice the granite, and these are microscopic. You cannot see these with the naked eye. They're very tiny. That's why he's sitting in front of his microscope there. And I'm going to give you just a little bit of nuclear physics so you'll understand how this worked. So please turn the lights on for a minute. And I need to go to this picture here. Now don't let this scare you. All this is a list of the elements. Hydrogen, helium, carbon, nitrogen, oxygen. In fact, if you took just those, the hydrogen, carbon, nitrogen, oxygen, you'd have 96% of your body weight. Everybody in this room knows a percentage of us is water. What is the percentage? You know what it is? It's actually 60% of our bodies are water, and that's I think you all know. I need a blackboard. It's H with 2 O's. You all know that, don't you? So if you add the nitrogen and the carbon, you got 96% of your body is just those 4 elements. It's amazing. your body weight. Now, I want to tell you something about this chart. Do you see that black number right there? In the corner of every box, whether it's calcium or nickel or selenium or whatever, that number tells you how many, now this is orange, but I'm going to call them pink. That number tells you how many pink balls are in the nucleus. I'm using pink because these are actually protons. You know that word, don't you? And they don't have little holes like this. I just had to buy something that I could use. Yeah. These aren't golf balls. These are practice golf balls. And so hydrogen, this is way too big. In fact, I told you if you scaled up a proton to be this big, the electron would be 6 miles out there. And 1/5 the size of a BB. But I don't have a small BB, so we're just, y'all with me on this. The idea is this is positively charged and this is negatively charged. So normally, and these things are flying around, that's a simplified version, normally the atoms are neutral. If you rip off the electron, which you can quite easily do, you now have something that has a +1 charge. Y'all with me? It's like static electricity in a way. So that's hydrogen. Now, helium is, what's the black number on helium? Clear over to the right. So you have 2 of these guys. Are y'all with me? I put a couple people to sleep, but that's not too bad. Let me show you another number. The number at the bottom of the box is how much the atom weighs. So if helium has 2 pinks and it weighs 4, that's because there's got to be 2, I showed you this morning, didn't I? There's got to be 2 neutrons, no charge, neutral. They weigh the same. And the electron is so light in comparison to, it's 1/2000th of the weight of one of these. The better term is mass, but weight is fine. So, if these white guys weren't there, what would happen to those 2 positively charged balls? They would fly apart. Y'all with me? So there's a force they call the strong nuclear force that holds these things together even though they want to fly apart. And so here's a very amazing fact. It's the number of these pink ones, they're not pink, but number of these pink ones that determine what the stuff is. So if I put another pink one here, and I might have to add a couple more white ones, what would it be? You see the 3 there? Isn't the 3 the number of pinks? Yes. And it's the number of pinks that makes it what it is. All of these elements are different because of the number of what? Pinks. Protons. Yeah. Amazing. And you have to have the neutrons in there or the babies will fly apart. Let's just take carbon since there's so much carbon in us. How many pinks in carbon? Can you read it from where you are? 6. And because it weighs 12, there's how many neutrons? Well, you know, whatever you want, you can have and make any atom you want. Are y'all with me on this? And you have as many electrons flying around as there are pinks. So the whole thing, if there's 20 pinks, how many electrons do you need? 20. So the whole thing is neutral normally. When you step back and look at it, if you got real close, maybe you'd see something flying by, but so forth. Okay. Now here's what happens. Oh, by the way, just for fun, all of these elements from plutonium on do not occur in the earth. Mankind has made these by firing inside of a nuclear reactor, firing pinks, and if they stick, you suddenly have a different element because it's the number of pinks that determines it, right? Should we all stand up and do some calisthenics to stay awake? I thought this was so fascinating nobody would ever go to sleep. You do as you like. It's like when I wake up at night, Neva reads to me, and I listen with my eyes closed, and I hear every word. And when I wake up, or when I open my eyes, I can't remember a word, so.
So I'm just trying to wrap my brain around this.
Yes.
So oxygen has 8 protons in the nucleus, and lead has 82 protons.
Yeah. Yeah.
So there has to be something different going on other than just the number of protons.
No, the number of protons determines what it is. You have to have neutrons there to keep it from flying apart. But you can change that. You can bombard it and knock some of them out, and then the element becomes radioactive. And I'll talk about radioactivity in just a moment. I just wanted to give you just a touch of background on how these elements work. And some of them are very heavy. By the way, hydrogen was the gas inside the Hindenburg. And tell me, how many times heavier is helium, which is in the blimps today, than hydrogen? No. 4 times as heavy. Hydrogen had 1. What does helium have? 2 whites and 2 pinks, but they're all the same mass. You all with me on that? Okay? In fact, if you take an electron out of this, you get a pink. That's crazy, but that's true. That's correct. To the number of pinks, if it's an unionized atom. Correct.
I'm having a hard time wrapping my brain around this. You have all these different elements just based on the number of protons.
Exactly. It's remarkable. Her statement is beautiful. All you have to do is add a proton, and you got a completely different element. I'm telling you, folks, chemistry in heaven is going to be fun because we'll understand everything instantly, right? You'll find me in the physics lab up there.
What happens when you apply energy or force to these things?
Let's not go there yet. Interestingly enough, as far as you and I are concerned, folks, we're just a few elements up here at the top, right? And some trace elements like some selenium or something like that, see, that our bodies use. Now, here's the interesting one, uranium. How many protons?
92.
Y'all with me? Yes, I'm sorry.
I can't understand it dim with
Oh, I should repeat the question. Yeah, I apologize. We should be using a microphone for the questions. If somebody wants to do that, for Lloyd's sake. How many protons, everybody?
92.
What's it weigh?
238.
It's the heaviest known element in the Earth. So they take uranium and clad tanks with it. Why? Because it is so dense, it's the best thing to stop some bullets or some missiles. You all with me? It's the heaviest thing that's out there. Now we've made heavier ones, but you can't. You can't make much of it. You make very little amounts. Okay, so 92 protons. How many neutrons? You'd have to subtract 92 from 238. That sounds like 38 plus 6 is 142, right? So 142 whites are necessary to keep these 92 guys from flying out. Y'all with me on that? Got it? Yeah. Crazy. Now, this all connects to the experiments that he did. let me show you another page now that you're blown away by this page. Actually, this comes in freshman chemistry in high school, and most of you did that. I'm sorry, I got to make these arrows move, so I'll just do it quick. Here's a very interesting chart. Do you see uranium-238? I should have had you just notice that thorium, where is thorium at? Should be right here. Yeah, there it is. Thorium has 90. Just keep that in mind. 90 reds or 90 pinks. So notice something. Uranium U-238, there's this alpha and an arrow, which means it's radioactive. And I'm sorry for the unusual things, but scientists long time ago decided to call a helium nucleus, are you all with me? An alpha particle. And what that diagram means is, got to trade hands here, an alpha particle comes flying out of the uranium and it becomes thorium, because how many reds, how many pinks did the uranium lose? What's its number now? 90. Got it? So uranium became thorium. Get ready, catch, because 2 of those guys left. Are y'all with me on this? Yeah, you're going to throw them back. And notice that thorium weighs 234, so it lost 2 of these and lost 2 of these. That's an alpha particle, isn't it? A helium nucleus. You all with me on this? Now, here's something very interesting. If you have a pound of uranium, every once in a while, it shoots off one of these. You all with me? And after 4.5 times 10 to the 9th, that's 45 billion years. 10 to the 9th is a billion. In 45 billion years, half of the uranium is gone. So the half-life, this is the term that scientists like to use, the half-life of uranium is 4.5 billion years. Are you all with me? You kind of know about that, don't you? Notice if you come down here to radium, maybe I'll just tell you this. Notice that thorium does not change its weight when it becomes protactinium, right? See, what happened was one of the neutrons became an electron and a pink. So the weight's still the same. But notice that the you don't see this there, but the atomic number went up. The number of pinks went up because one white one became an electron, and the beta on there is all that is an electron. How much less does an electron weigh than one of these guys? 1/2000th. Remember that? It's a little different than 2,000, but it's very close to 2,000, so we just say 1/2000th. So the weight hasn't changed. You see that on the screen? The total weight is still what? Same weight. It's just that there's now one more pink, correct? And so that happened because the neutron shot an electron out, and now it's pink. It's positive itself. Same weight. And here's the interesting thing about that. If you come down to radium, I'm sorry, radon. Radon is a what emitter? Alpha, which is helium, right? Now, that alpha particle can't even pass through paper. And it can't even pass through your skin. So it's not harmful. But when you get it into your lungs, that alpha particle can damage cells because the cell membrane is thin enough that it can go right through it. 10,000 people a year die in the U.S. from radon poisoning every year. Doesn't hurt you from the outside. Y'all with me on that? Y'all with me? But breathing it. So all of you know about that. You're supposed to test your basement. Now, if you have a door in your basement that gets opened very often, radon gas is really heavy. It just flows right out the door. And if you have windows in your basement and they're cracked, it'll flow right out the window. But from the window on down will be what? Some radon gas, see. So now, back to Bob Gentry. That's enough nuclear physics for dummies, right?
Quick question. So why does it go, you have uranium is 4.5 billion years, and thorium's half-life on 24.5 years.
Oh, so the question is,
And then 1.14 minutes. And
yeah,
then it starts.
yeah. So the question is, why are the half-lives so different? And that's not fully understood, but it has to do with how many pinks and whites, and it's different. And what Robert Gentry found, listen closely to this now, he found that there were some radioactive spots in granite that had half-lives of seconds or minutes. Don't go, just follow me. He found some radioactive centers that had very, very brief half-lives. Now follow this reasoning. This is beautiful. He wrote a whole book on this. If the granite, as the standard geologist believes, they have no idea how this actually happens, but they believe that all these rocks formed from a volcanic melt over millions of years. If the half-life of that spot that he found was short enough, all of it would have been gone by the time the stone formed. Because its half-life is so short. Y'all with me on that? The fact that he found those in granite that had such short half-lives, found the rings. You can, with teaching and skill, you can tell which ring was caused by what element. Are y'all with me on that? In order for that granite to have formed and still preserve the half-life marks of a short-lived, a short half-life, the granite had to be formed instantaneously. That is to say, it had to be created in an instant. Are y'all with me on the idea? So the name of his book was called Creation's Tiny Mystery. And even the atheists, even the evolutionists say, yeah, you got an interesting point there, Gentry, but we don't believe it. And unfortunately, Bob he was my teacher in my freshman year, algebra, calculus, and so forth. And he was only 5 years older than I, or so. And we became friends. And when he wrote the book, he sent me a copy, and he said, please proofread it for me. So I went through the whole book, and he's a good enough physicist, but he thought maybe I could find something. But anyway.
Hey, Jim.
Yeah.
Is that all granite or just certain types of granite?
I think it's all granite, but that's a good question I never thought about before. Unfortunately, Bob, oh, I love this man. His daughter is Patty Guthrie. You know the Guthries, some of you, don't you? Patty and George? No, Patty and Todd Guthrie that live in Shasta. She was a 1-year-old baby girl when he was teaching me college algebra. But anyway, I don't know how this happened. People started attacking Bob, and instead of kind of, I don't know, going with the pressure, he would kind of get testy. And he got so testy that it just wasn't useful anymore to have him talk to groups, and it was a sad story. I went to see him before he died a couple of months. We heard that he was ill, and I didn't know he'd remember me. Oh, man, he grabbed my hand, you know. And so that's probably enough. But this is the chart that shows the decay of uranium. And it's not very interesting to most people, but I thought it would be interesting if you understood radon, and I thought it would be interesting if you understood how Bob Gentry's work showed that creation made these things in an instant. What does the Bible say? He spoke and it was done. And I think we're done. I've got more pictures, but we should quit. Oh, yeah.
Radon is heavy gas, you said.
Yes.
Then when they build a house over radon, they have to put a pipe in.
Yes.
With 6 inches of gravel and a film of
visqueen
plastic. Is
let me describe it. So in order to mitigate is the word they like to use. When you get a permit today, and somebody told me, I don't know if this is true, that years ago there was a restaurant in Kettle Falls called the Geiger Counter. Because you may not know this. This is a hot area in the country for uranium and radon. And so you can't get a building permit to pour a slab for a basement. It's okay if your house is elevated with a skirt. That's okay. But if you've got a slab in your basement, you have to put down pipe, all interconnected, and finally a pipe that goes outside, and then washed pea gravel, and then visqueen, and then concrete. And then you put it, and then you test your basement when the house is built for radon, and if it's present above what's normally outside, which is it's there all the time outside, then you put a fan on that thing that sucks on that pipe 24/7, and it clears the radon. And that was the case when I added to our house. I put a big slab in the basement, and the radon was high. Turn that fan on, and in a day or two, the radon level is the same as it is outdoor. So it does work.
Put a fan in that pipe.
You have to put a fan in if you find radon. If you don't find much radon, you don't need a fan.
Oh, okay.
But the fan, all these pipes are connected to one pipe that goes outside, and that's where you put the fan. So, anybody else? I'd love to talk to you about atomic and nuclear bombs, but I suppose we should quit. Let me close with this amazing picture. You remember what this is? Just for the sake, see, I don't have a blackboard. This is the BRCA1 protein, BRCA1, when they found there was another one, BRCA2. And they thought it was breast cancer in females, but it turns out to be all female reproductive organ cancers and male reproductive organ cancers. And if the BRCA1 or 2 gene is mutated, then that thing can't fix the mutations that occur in people's copy error, right? Or in outside radiation. And most of us are not moviegoers. I had no idea who Angelina Jolie was. I never heard the name before until she found out that the mutations in her BRCA1 and 2 genes, which she inherited most of them inherited, the mutations were substantial enough that she had an 87% chance of getting breast cancer instead of the average American female 11%. So she had surgery. That's when I learned about who she was. So this idea of knowing what mutations are in your genetic background is becoming a big issue with not just diagnostics, but prevention. If there is something that you could do ahead of time. But it's just like I told you this morning, John, and I looked up his name and now I can't remember it, the author of the book, Genetic Entropy. There is so much mistakes happening in DNA that if it wasn't for these tumor suppressor proteins, everybody in the world would die within 40 years from cancer. Isn't that amazing? The problem is they miss some, so we still have problems. Let's pray. Again, Lord, we thank you for the marvel, oh my Lord, the marvel of your hand. And we scratch a little bit for some understanding. And it just makes us love you more, appreciate you more, want to take better care of what we have. And bless our people, Lord. Bless our church, Father. Our church is struggling with itself these days the world over. It's a heartbreak to see how the shaking has occurred in a way that people don't necessarily leave the church. They're shaken out while they're still in the church. So help us, Lord, I pray in Jesus' name. Amen. Well, thank you for hanging in there. I hope